nature, the shape of the tiny pipes corresponds exactly to the requisite flow characteristics. So even oily and watery liquids can mix, and the mixer can be used precisely where it's needed. So the transport of dangerous chemicals is largely avoided. Chemicals are still produced worldwide in a complex jumble of installations. One minor defect and an uncontrolled chain reaction can endanger the public and the environment. But with microchemical production, many small reactors manufacture the same materials. Their joint output is equal to that of big plants. However, should something go wrong here, the consequences are containable. Mixing on the micro level. New opportunities for manufacturers, drop by drop. A chemical plant in the smallest of spaces. Will this make the threat to the environment from large factories a thing of the past? Is there an even smaller dimension? Yes, nanotechnology, the 1,000th part of a micrometer, the millionth part of a millimeter, the realm of molecules and atoms. The invention of the scanning tunneling microscope by Gert Binnig and Heinrich Rohrer made a journey into the tiniest dimensions possible. Just a few atoms wide, the tip of a metal probe scans the surface of a conducting material without touching it. A minute current flows between the two, tunneling current, which varies according to the distance. A feedback loop keeps the tip at a constant distance above the surface. In this way, the scanning tunneling microscope provides an atomic scale topographical image of the surface. The image forms line by line. Individual atoms become visible, arranged in weird and wonderful patterns. This technology provides scientists with an insight into undreamt of dimensions. The nanocosmos has become accessible to them. Lennart Nielsen perfected this microscopic technique at an early stage. He uses it to study human cells. White blood corpuscles combating a virus appear to him like an array of stars.
More or less the damaged white blood cell tremors.
When they've been stained, even dangerous pathogens seem strangely fantastic. A white blood corpuscle attacking bacteria. The AIDS virus. A malaria virus destroying a blood cell. A journey into the interior of a leaf. Through insights like this, we see the wonders of nature in a new light. Today, scientists are able to explain many of nature's phenomena, like photosynthesis. One variation of a scanning tunneling microscope, the atomic force or scanning probe microscope, can even manipulate individual atoms. A force is created between the tip and the sample. This force is sufficient to displace individual atoms. This possibility has encouraged many researchers to think seriously about a whole range of very unusual concepts. Could such technologies perhaps rearrange individual atoms to form miniature machines? Or could matter even be formed directly from its atomic building blocks? Perhaps one day we might each have our own private matter generator with which we can furnish our home just as we wish, completely new every day. Waste disposal problems would be a thing of the past. After all, atoms would merely be rearranged. Shapes, colors, everything. However, influencing atoms or molecules is a massive undertaking. incredibly sophisticated technology in ultra-clean rooms is needed to move just a single atom, let alone thousands of billions, as in this case. In their boundless enthusiasm, many had forgotten that visions like this are merely concepts to be mulled over. In the meantime, serious scientists have been working to clarify the true potential of nanotechnology. Some work here at Keio University in Yokohama. They're studying long chains of molecules, like individual chains of DNA, for instance. These can only be removed from the cell system in lengthy, monotonous chemical processes. Living cells are natural molecular machines. This scientist is going to demonstrate something quite fascinating. Using fine beams of light known as laser tracers, which she adjusts very carefully, she grasps the ends of the chain of molecules. Amazingly, she ties a knot in it. Professor Kazuhiko Kinoshita explains the true motive behind this research.
For us, it's just for fun, and actually we are very impressed by the this miraculous molecular machine that Mother Nature has produced, and we would like to know how this machine operates. We want to know the mechanism, and of course other people can think about applications, and that's not what we are going to do.
Professor Eberhard Unger from Jena in Germany also depends on nature's help in his research.
We need organisms that produce these building blocks very cheaply and in large quantities, in particular in comparable and standardizable quality. It's essential for the systems which operate in these tiny dimensions to be easy to control and for them to function consistently.
Researchers at the Institute of Molecular Biotechnology in Jena obtain their building blocks from pigs' brains provided by the nearby abattoir. Two specific types of molecule occur in brain cells with particular frequency: microtubules and kinesins. In the brain, microtubules and kinesins are responsible for transporting messenger substances and nutrients between individual neurons. In an intricate process, scientists are able to remove these cellular building blocks from pigs' brains. But the two molecules can also be obtained from plants and microorganisms like bacteria. Countless biological separation processes are needed. It's a case of centrifuging, then adding chemicals and centrifuging again. But in the end, scientists obtain a supply of the precious substances. The microtubules and kinesins can now be brought together in a suitable way. It's vital to find the right mix. Under the microscope, a remarkable world reveals itself to the lab assistant. Kinesins, tiny dots moving over the microtubules like trains on tracks. Powerfully magnified, the microtubules resemble a rail network. The simple principle involved, the kinesins virtually walk along the microtubules. Where applications are concerned, the right mix is crucial. More or less of the two basic building blocks, and the researcher obtains a new product. Here, kinesins are transporting larger molecules. When several kinesins adhere to a base, the microtubules migrate on them, almost like tiny worms. Professor Unger.
Also diese Arbeiten
At
sind
present, we're at the preliminary research stage. We plan to find out whether there's an advantage in powering nanomotors or nanotechnological devices with biological drive systems of this type.
Antriebssystemen vorteilhaft betrieben werden können.
If such machines are to transport specific substances one day, a direct external influence will be needed. In this case, a special liquid flows over the microscope slide. As a result, all the microtubules become aligned in the same direction. But these initial successes also show how far science still is from building an artificial molecular machine. Will it ever be possible? Professor Kinosata.
This is a machine that Mother Nature has made, or God has made. I don't think that human being can produce such a tiny machine that really works.
For years now, nanotechnology has achieved undisputed success in the field of electronics. Its influence is now an integral part of everyday life. For instance, countless sensors play an important role in car manufacture. Without them, automotive safety systems just wouldn't operate. The electronic system has to register an accident and inflate the airbag in a fraction of a second. Other forms of microelectronics make modern data exchange possible. Through the internet, huge volumes of information pass from one end of the world to the other. Modern medical technology, studies of new surgical techniques using virtual instruments. Nanotechnology has become firmly established in many areas of research. The Fraunhofer Institute in St. Ingbert in the Saarland, just one of many examples. Scientists here are already practicing what in future will become standard, the merging of different specialized fields. Up to now, pharmaceutical researchers have had to examine the effect of active agents on cell cultures by means of biochemical analyses, a laborious process. But here, a scientist merely places a cell culture directly onto a special microchip. Each cell generates its own electrical impulse. The microchip can measure this directly and record it. Biology combined with microtechnology, that's the strong point of this research. It permits faster and more accurate testing of active agents in the development of drugs. And advances have also been made in cancer research, in isolating defective genes. In future, there might even be drugs tailored to each individual patient. If biochips can measure the characteristics of cells, couldn't electrical impulses selectively stimulate individual cells? That's a prerequisite for the development of artificial cells, like an artificial retina. One day, scientists hope they'll be able to implant these chips and help blind people to see again. Initial tests are already being carried out on pigs and rabbits. This rabbit is being fitted with a sensor, rather like a contact lens. The animal is kept under anesthetic throughout the experiment. A new series of tests gets underway. A flickering light stimulates the eye. A computer measures the reaction via an electrode under the skin of the rabbit's head. But what do such experiments with animals actually tell us? Professor Eberhard Strenner from Tübingen University Eye Hospital.
Natürlich könnte uns ein Mensch.
A person, of course, could tell us what he or she can see. But the problem is that we can't take any risks, even with people who have lost their sight. The experiments might lead to inflammation and so on. Above all, we have no idea how the chip would be tolerated in the long term, how the eye will react over a longer period.
Auge einen solchen Chip.
Or how a chip of this kind would tolerate the eye. Here, a scientist compares a chip that's already been implanted with a new one. The surface has been damaged, as indicated by the discoloration. This must not happen when a chip is implanted in a human eye. And nanotechnology, it's hoped, will prevent that. In a vacuum chamber, researchers vapor coat different layers onto the retina chip. They hope one day to find the right combination of materials. But it will be a while yet before blind people really are able to see with artificial eyes.
Wir sind ja auf einer Art Expedition.
We're on a kind of expedition into uncharted territory, where we can expect to encounter difficulties we can't at present envisage. The early explorers in Africa never knew if they were going to come across an impassable mountain range, and that's the sort of position in which we could find ourselves.
Auch noch gehen.
The expedition into the tiniest of worlds advances further and further into the wonderful world of atoms. New insights into an amazing dimension with breathtaking potential. Many discoveries have been made already on the journey into the very heart of matter. Nature often shows us the way. Why is a lotus leaf always clean and dry? Dirt doesn't stick. It's simply soaked up by water and flushed off. The secret is revealed once again by the scanning tunneling microscope. The surface of this velvety soft leaf is not smooth, but consists of countless tiny elevations covered with the plant's own wax. Even adhesive doesn't stick. The technical replication of such structures paves the way for numerous applications. It makes separating compounds superfluous, so there are fewer waste products in the production chain. When nanoparticles are vapor-coated onto the right-hand side of a mirror, it no longer mists over. The pane on the right has been coated with a nanostructure. Dirty water doesn't stick to it. The big question is whether such nanostructures can be manufactured in large quantities and not just in the lab. If so, in future, industry will be able to use far less water and fewer chemical detergents, but such developments cannot be achieved overnight. Professor Kurz from Aachen Technical University.
Wir müssen heute forschen.
We have to perform research today in order to develop something that will be topical in 15 years' time. The more complex the field, the longer the research. And this automatically means that more time is needed to penetrate new dimensions.
Automatisch.
In future, governments and industry will have to invest a lot more in basic research, because only by understanding the atomic structure of matter will scientists be able to devise tailor-made products. Take steel, a material that requires complex finishing. Soon perhaps it will be ready cast or stamped to suit customer requirements. But whether we're talking about jet engines or ordinary car tires, research from the fields of micro and nanotechnology is behind many industrial products. But are there no limits to this research? Can our knowledge keep on increasing? Professor Unger from Jena University.
With unlimited financial and other resources available, yes, I think it could. But since that's not the case, in the new millennium, science will focus primarily on how to make more food and water available.
Research in the new millennium. A shortage of raw materials will force even the affluent society to reflect once again on people's basic needs: food, water, and above all, energy. Nanotechnology can help to improve the efficiency of solar cells and their range of application, so that the use of fossil fuels and the consequences of this can be reduced. The world at the turn of the new millennium. What role will science play?